In this video, I will be going over Lab 1 on blood. Remember, your labs are located in Connect, and you're going to access them through your Brightspace homepage for the course. So you'll find in there ID list, click on those, and those will take you into images in Connect. You'll be able to go through all of the structures that you need to be able to identify. And there will also be some text-based information. In addition, I may at times post histology images in Brightspace in a PowerPoint. So you can refer to those as well. Those are going to be different views and they might be helpful for you. So let's begin. When you first go into the ID list, this pop-up window will become visible and you can read through this. You can learn how to pronounce the terms. You can also, sometimes there will be videos there and so forth. And you can look at the structure list and layer tag button is also available. So I'm going to get rid of that. We're going to start with the list here. This is my first activity. What I've done is I have broken down each lab into multiple activities. And what I would do is I would go through the first activity as often as you need to until, well, you can go from activity to activity, but you can repeat an activity until you have all that information down. And then you can move on to the next. Your quizzes on this material, or your quiz for this week on this material, is going to open up on Monday. After you go through this information, you learn this stuff, you can take the quiz and you can take it as many times as you want to, and your highest grade will count. But remember, once the due date is there, you cannot take it anymore. So your due date is when you have to submit it by. So we're going to come over here. You can see these are the structures that I'm going to ask you to identify in this slide. For blood, and you'll need to go through your worksheet for this lab as well. There'll be some text-based information in there that I might ask you about. But for blood can be divided up into plasma and formed elements. Plasma will be the fluid portion of the blood. It's a straw-colored fluid, and that's going to be what looks like white here. So the background. You're going to have a variety of things dissolved in the plasma, and you can look at the lecture material in your textbook to find out more about it. The formed elements are the cells, and they are going to consist of erythrocytes, which is another name for red blood cells. You'll also have leukocytes, which are your white blood cells, and you will be learning 5 different types of leukocytes. You'll have to be able to identify them, tell me what their function is, and whether they are what we call a granular or a agranular site. So a granular site or an agranular site, and I'll talk about that in just a minute. So you'll have leukocytes. Neutrophils are the most abundant of the leukocytes. The way that you can remember which types of leukocytes are the most abundant to the least abundant is a mnemonic, which is never let monkeys eat bananas. And the never stands for neutrophils being the most abundant. L for lymphocytes, M for monocytes, E for eosinophils, and then B for basophils. So basophils are going to be the rarest, which means that in a blood smear, you're going to see very few of those. The third type of formed element is what we call a platelet. It's important in blood clotting. The thing to remember about platelets is that they are not entire cells. They are actually fragments of large cells called megakaryocytes. Mega means large, karyo refers to the nucleus. So let's begin here. When you look over here on the left, you'll see what looks like a clipboard. That's always going to give you the structure list, which is what we're looking at here. If you go down here where you see the eye, that's going to give you some information about what it is that you're looking at here. So I want you to know, let's click on this eye. I want you to know that this is a blood smear. Should I ask you on the test, what is this an image of? It is a blood smear if I'm talking about the entire slide. Let's start with the erythrocytes or red blood cells. Now, when I click on this, notice that all of the red blood cells or erythrocytes are highlighted. Let's say I don't like that color. I can go over here to the eyedropper, click on it, and I can change the color. So I'm going to change it to yellow. Well, maybe after I know where the cells are located, I want to get rid of the highlights so that I can actually see those cells or that structure if you're looking at the cadaver. So I can click on what looks like a highlighter and it goes away. If I need to get some information, and you will, I want you to do this with all of the formed elements. I'm going to come over here to the left and click on I up here, and that's going to tell me about erythrocytes. Well, let's say I don't know how to pronounce erythrocytes. I can click on this.
Erythrocyte.
And then if I want to go to a video or an animation, there is one on hemopoiesis, or the same thing as hematopoiesis, which is something that we're going to cover in lecture. So you could watch that video for lecture. And that's blood cell formation. So here's information where you find erythrocytes, what their shape is, and they're shaped like a biconcave disc. A disc is a circular structure like a flat ball. Biconcave, bi means two. It means that it caved in on both sides of that flat, round structure. And where it caves in, it is lighter in color. So you see that it looks white there in the center. What happens is before the erythrocyte moves from the red bone marrow into the blood, it ejects its nucleus. Once it ejects the nucleus, it collapses where the nucleus was. So the nucleus would have been here in the center. It collapses, and that creates that light colored area. Think about a donut, but instead of having a hole in the middle, it's very thin. That's what it's going to look like. So that would be the description here. If I read this, it tells me that erythrocytes make up about 99% of all the formed elements. So when you're looking at a blood smear, if it's a normal smear, most of what you're going to see of the formed elements are going to be erythrocytes or red blood cells. And you can see that's kind of true. These large cells here are going to be leukocytes or white blood cells. And you can see the dark staining structure there in the center. That is the nucleus. So the shape of the nucleus is going to tell you what type of leukocyte it is. And we'll talk about that in just a minute. So that is going to be a leukocyte. And all of these are my erythrocytes. These little specks, remember, platelets are fragments of larger cells called megakaryocytes. So these little flecks here are all of your platelets. And here I have a clump of platelets. So what is the function for an erythrocyte? Again, I might ask you to tell me this on the quiz and exam. It is to transport oxygen and carbon dioxide. As a matter of fact, 98.5% of the oxygen that is being transported by your blood to your tissues so that they can use it to make ATP is transported inside the red blood cell, bound to a pigment, a protein pigment called hemoglobin, and we'll look at this in lecture. Also down here at the very bottom, there might be some additional information. In this case, it's telling you that an erythrocyte is also a red blood cell or an RBC. Now on the quiz, you have to write it out. You can't use any abbreviations. So now we're going to go on to the next structure in that structure list, and that is my neutrophil. So notice that these white blood cells here, these leukocytes, are highlighted. Those are my neutrophils. Remember the mnemonic, never let monkeys eat bananas? So I know my neutrophils are the most abundant. And what tells me this is a neutrophil, and they're really easy to identify, is I look at the nucleus, and you can see that the nucleus is pinched into lobes. A nucleus will be pinched into 3 to 5 lobes. So I can see one, that's possibly 2, 3, 4 right there. So they're going to be the most abundant of your leukocytes in your blood smear. And what happens with leukocytes, the reason you don't have many of them in your blood is due to the fact that they leave the blood And they're going into other tissues so that they can do their job. They're going to be part of your immune response, and they'll be really important in that. So if I go back over here to neutrophil, so remember I can
Neutrophil.
play the pronunciation, the location. They will be found in the blood, but they're also going to be found in your tissues. It's going to give you a description, the 3 to 5 lobes. That's really what I want you to know here. Don't worry about the staining. We don't talk about that. But another thing that you need to know with all of your leukocytes is that you're going to have 2 main groups of leukocytes. Those whose granules, and granules are structures that store different types of chemicals. And these are chemicals that they're going to use when they're trying to get rid of a pathogen or carry out their immune response. So in those granules, if the granules, when you stain the cell, are visible, and you can see it's kind of grainy here, these cells are going to be called granular sites. So I don't see it here, but we'll see it in another slide where they talk about granular sites. But again, you're going to have this in your lab worksheet, so be sure that you read about it. If you have a leukocyte that when it is stained, the granules are not visible, they're still there, but you can't see them, they're called agranular sites. And remember, the prefix A means without. So they're just not visible when it's stained. A good rule of thumb is that if the leukocyte ends in PHIL, Phil, so you can see neutrophil, it will be a granular site. If it ends in cyte, CYTE, it will be an agranular site. So if you just remember that rule, phils are granular sites, cyte are agranular sites. So this is going to be my neutrophil, and you just need to know what their functions are. So I want you to be able to identify it. You're not going to have to tell me you have a nucleus with 3 to 5 lobes, but you will need to know that to identify them, that they are the most abundant. And I want you to know the function. They are phagocytic, which means that they're going to eat bacteria. They are the first leukocytes when you get a cut and bacteria go in across your skin, underneath your skin. They will be the first leukocytes there. They're going to be eating the bacteria. They also use other types of immune responses to get rid of the pathogen. And I do have some videos in Brightspace, so go ahead and watch those videos so you understand what they do a little bit better. Let's move on to the third one on our list here, and those are the platelets. So again, remember I said these are the little specks. Let's remove the highlight. So again, that was the, oops, I don't want to change the color. That was the highlighted platelets, and there you can see them more clearly here. So what do platelets do? Platelets, remember, are fragments of larger cells called megakaryocytes. Here we go. And in their cytoplasm, they are going to have chemicals that are going to be involved in blood clotting. So it's going to come down here to the function, contributes to clot formation. Also, they're known by another term, thrombocytes. And you do need to know that term because we're going to talk about thrombocytosis, thrombocytopenia, thrombopoiesis. So if you know that a thrombocyte is a platelet, that will help you remember what it does. Now, we've gone through this list. I can go through it as many times as I want to, and then so I can go back, use the back arrow here until I can, at this point, identify everything until be able to describe what they do. So remember, this is an erythrocyte, this is a neutrophil, and these are all platelets. Now I can go to the second activity since I've finished that. And I want to know what this is an image of, photograph of. So I'm going to go down here and I'm going to click on the eye for the image description. And it's going to tell me that it's a capillary. And inside the capillary are all of these erythrocytes. So you can see how they're caved in there. So biconcave disc, and remember, they're transporting respiratory gases, oxygen to your tissues, carbon dioxide to your lungs. So let me highlight the capillary. There's the capillary. And I can highlight the erythrocytes within the capillary. These here, these cells, they don't identify those, but those are different types of leukocytes. So kind of cool here that you can see these. I don't know, is that a word that you use anymore? So if I go here, this is going to give me the same information about erythrocytes that I saw before. So now I'm going to go to the third activity. What I've done here is you can see an enlarged image of this cell here. So if you look at it, remember the rule, if it has 3 to 5 lobes in the nucleus and the dark staining structure is the nucleus, it is a neutrophil. So this is going to be a neutrophil, really easy to identify. And you should be able to tell me, is this a granular site or an agranular site? Remember, all your fills are granular sites, and you can kind of see the granules here. Do you recognize these? These are your erythrocytes. Whoops, since I didn't complete. Oh, here we go. So we're highlighting here the neutrophil. Now we're going to go to the next one. So this, what I want you to notice, you're going to have kind of reddish granules here. That I think is probably the most characteristic attribute of these cells. These are called eosinophils. So eosinophils, let's click on that. I'm going to take the highlight off so that you can see it. I'm going to click on the information. Let's say I'm not sure how to pronounce it.
Eosinophil.
Click on that. And then the description here is going to tell you that the nucleus is going to have 2 enlarged lobes. This is actually one lobe, and this is another. So it's a little confusing. You might confuse this with a neutrophil, but the neutrophil will not have the red granules. So let me get rid of the highlight. See there? So that's going to be the difference. Plus, it's going to have more lobes. But this is actually one lobe here, and this is one lobe here. It's just that it's partially covered up by the granules. You're going to come over here and you're going to look at the function. And again, you need to know this for the quiz and the exam. They are going to do 2 main things. Their numbers are going to increase if you're having an allergic reaction and if you have parasitic worms. So go ahead and read that information here. And remember, never let monkeys eat bananas. So they're not the rarest, but they're second to the rarest of your leukocytes in your blood. Now we're going to go to the next one. I like to describe this as a fuzzy ball. Usually you can't even see the nucleus, so I don't really worry about the shape of the nucleus because I can't see it and it's not going to help me identify it. But if I see a fuzzy ball, that is going to be a basophil. So I go over here. What is a basophil? The function is that they are going to be involved in initiating inflammation. Their granules store histamines and heparin. And when we talk about inflammation, once we get into the immune system, you'll learn that inflammation is a response to tissue damage, and that damage could be due to multiple things. It could be due to an infection, but it could also be due to chemicals, burns, whatever. So read this and remember that they are the rarest, less than 1%. You don't have to remember these percentages for the individual types of leukocytes. Let me go back. I'm not looking for the percentages. I'm looking for the number of cells in a millimeter cube. So go to your textbook, learn how many cells, what's the normal range for erythrocytes in a millimeter cube of blood for leukocytes all together and for platelets. But don't worry about the percentages, just know what's the most abundant, what's the least abundant, and remember the pneumonic. Going on to the next one, the lymphocyte. So notice with the lymphocyte, do you see this little, what I like to describe as a halo around the nucleus? So this is all the nucleus is really large. See how that is similar to the basophil? To be able to distinguish these two, what you need to look for is this halo. If you see a halo of cytoplasm, and that's what it is because this is the nucleus, then it's going to be a lymphocyte. And lymphocytes are involved in your specific or adaptive immune response. All of your leukocytes are part of your immune response. Lymphocytes, though, are part of your specific or adaptive immune response, and we'll learn more about what that is. So when you give me a function, you have to say it's specific or adaptive, since saying it's just its function is your immune response, that's too generic. So let's look at some information here. It's going to give you some information where you find it. It's going to tell you that you have B lymphocytes, T lymphocytes, and it talks about natural killer cells, which the natural killer cells are actually not part of the adaptive immune response because, or specific, because they are non-specific. So we'll talk about those again. And it's going to talk about what the function is and describe a little bit more about it. So be sure that you read this. Again, I'm not going to ask you for the percentages, so don't worry about that. Just the total number of leukocytes, normal range in a millimeter cubed of blood. So now we're going to go to the next one. This one here, this is a monocyte. Monocytes are the largest of your leukocytes. See how much bigger it is than your erythrocytes? And what you need to notice here is that the nucleus is going to be shaped kind of like a kidney. So it's not pinched in here. And even though this looks a little grainy, remember all of your sites are agranular sites. So this is going to be an agranular site. Let's look at the information here. It's going to tell you that they are phagocytic cells. As a matter of fact, when they leave the blood vessel, they are known as macrophages, and macro means large. A phagocytic cell, phage means it's eating something else. So these are going to be your macrophages once they leave the blood. So go ahead and read this information. You see their numbers increase when you have a chronic infection. Here, it describes granular sites and agranular sites, and it tells you which type of leukocytes fall into each one of these categories. I'll ask you what class these cells are, and you're going to have to tell me they are granular sites. And the same thing for the lymphocytes and monocytes are agranular sites. Moving on. You'll have to tell me what this is. Well, let's say I don't recognize it. Hopefully you do from AP1, but if you don't, you're going to go down here, click on the image description, and it's going to tell you cardiac muscle. If you recall from AP1, cardiac muscle is found only in the heart. Cardiac refers to heart. So I'm going to put in the information here. Found only in the heart. It is striated, so you can see the little stripes here. The other type of muscle that is striated is skeletal muscle. So it's striated, and that's due to the arrangement of the thick and thin filaments. It is branched. So I'm going to see if I can see, you can see how this is branched right in here. That's going to be important to the function of the heart, and I'll talk about that when we get into the heart chapter. Chapter 19. And if you recall, there are special junctions in between adjacent cells, and they stain dark. There's one there. And those junctions are called intercalated disc. We'll talk about the membrane junctions that you find there, but those are going to be important as well for the heart to function properly. So I can go over here and I can read about it. Remember, cardiac muscle is involuntary. So this is going to be found in the myocardium of the heart, which is the muscle of the heart. Let's go to the next one, the intercalated discs. So you can see here they're highlighted, and it'll give you a little description about them. And then, wow, I don't know what this is. I'm going to go down here, click on image description. Is going to tell me I'm going to have an artery and a vein here. Well, how do I know which one? This is the lumen of the blood vessel. The lumen, if you recall, is the hollow portion of the organ. This is where the blood is going to go through. So this is all blood. You can see all these erythrocytes. So here's the lumen as well. These two are not of equal sizes, but see how the lumen here is nice and round? You can see the blood there. See how it's partially collapsed here? I need to pay attention to that. If it's nice and round, it is an artery. If it is partially collapsed, it is a vein. So you're going to have to tell me whether it's an artery or a vein. You also can look at where you're going to have to be able to identify the 3 layers of the vessel wall, and we'll look at that in just a minute. But there are 3 layers. We call them tunics, an inner layer, a middle layer. You can see this middle layer here, and an outer layer. So I'll get to that in just a minute. But notice that this middle layer here is thinner than this middle layer here. So arteries have a thicker middle layer than veins do. So let's go to, I believe this is the last slide. This is a magnified view of an artery. And the reason I can tell it's an artery, and by the way, I could go here, click on this, it's going to tell me it's an artery. You don't have to worry about the elastic part or the muscular part, but this is an artery. I can tell because if you look here, this is your lumen, the hollow portion of the tube. So we're going from inner to outer. Notice this light staining layer. That light staining layer is called the tunica intima. And notice how it's scalloped. So when the artery is dilated, the scallops can disappear. When it's contracted, you see the scallops appear. So this allows for vasoconstriction and vasodilation. So we're going to go here. I have a description of the tunica intima. It's called intima because it's in contact with the blood, like intimate apparel is in contact with your skin. And if I look over here, down here at the comment, it's going to tell me it's also known as the tunica interna. You can use either term because it's the innermost layer. So that's here. It's going to describe what it consists of. So an endothelium, which is simple squamous epithelium that you find only in the cardiovascular system. It has a basement membrane. Now that basement membrane for arteries will be present. When you're talking about capillaries, it may or may not be present, or it may not be complete. In capillaries, that's all you're going to find is this tunica intima. But for your other vessels, you're going to have the 3 tunics. So read this information. It talks about vasoconstriction and dilation here. So again, this is going to be, it is vasoconstricted. The tunica media, as we're moving out, that's going to be this highlighted area here. Let me remove the highlight so we can see it here. If you recall from AP1 what smooth muscle looks like, this is smooth muscle. This tunica media then consists primarily of smooth muscle, and it is responsible for vasoconstriction and vasodilation. When you have vasoconstriction, the lumen becomes smaller because the smooth muscles are contracting. When you have vasodilation, the lumen becomes larger because the smooth muscles are relaxing. Recall, arteries have a thicker tunica media than veins when you're comparing the same size vessel. So tunica media here. So go ahead and read about that. And then we have, tunica externa. So I'm going to go, here we go. There's the tunica externa, and that's the outermost layer. So we're going from tunica intima or interna. This layer, remember, is tunica media. And then the outermost layer is the tunica externa. It's going to tell me it's going to consist of elastic and collagen fibers. It's like putting a stocking around the blood vessel, and this prevents blowouts of the blood vessel. So if you have really large arteries and veins within this tunica externa, you're going to have blood vessels as well to supply nutrients and oxygen and remove waste products from that large blood vessel. So that's called a vasa vasorum. And that'll be, I believe, in your, here we go, vasa vasorum there. And that, and you'll have small nerves in this area as well, supplying the smooth muscles here for the vasoconstriction and vasodilation. Also, the tunica externa is known as the tunica adventitia. So you have two different names for this layer here. So remember, the layers are called tunica, and you just need to be able to identify the layers. Tell me something about the layers. And I believe that, no, we still have a few more slides. Oh, so I think this is kind of cool. This, well, let's say we don't know what it is. I'm going to go down here, click on the image description. It's going to tell me it's, you're going to have blood inside a blood vessel. So what do you think these are? These are all erythrocytes. Now see this? Do you see how it has a dark staining nucleus? That's going to be a leukocyte. You can't really see it well enough. It's a neutrophil, but I would never ask you to identify this as a neutrophil because you can't see it well enough. You're going to have the enlarged images to identify when I'm asking you to identify the different leukocytes. And on the quiz, when I ask you to identify a formed element, it will either be an erythrocyte, some type of leukocyte, or a platelet. Whoops, did I? Okay. You want me to click on the erythrocytes. All of these are erythrocytes. Now we're going to go. So hopefully this was helpful. You need to go through this week. Your quiz will open up Monday, and you'll have until Sunday at 11:59 to submit your quiz. Now, you can take it as many times as you want to. I'm going to encourage you to take it over and over and over again so you can get used to taking a quiz, because your exams are going to be in the same format. So take it as many times as you can. You're going to be typing in the correct answer. It's in lab when we meet in person. You have to write the name down. It has to be spelled correctly to get credit. So you're going to type it in. Be sure you read the directions before you start. Type in your answers. You will have 40 seconds for each question. I hope this was helpful, and I will see you in class.